go through the lecture questions for that topic, and then we'll get started with photosynthesis. And I just want to get through half of photosynthesis, and then we'll stop there. Okay, so the first question, let me make that larger. Okay, does anyone remember, because this was a little bit ago, how a cell detects a signal? So we talked about the cell signaling was important because it allows either two cells to communicate with each other, and they're going to do that through signals, or it allows the cell to respond to signals in the environment. What part of a signaling pathway allows it to detect the signal? Receptors, so just receptors, yeah, just receptors. So receptors is what I was looking for. And if you just remember back, we talked about 3 different types of cell surface receptors. We talked about enzyme-linked receptors, G protein-coupled receptors, and then also ligand-gated ion channels. Okay, does anyone remember then what a second messenger was with respect to cell signaling? Okay, so just to review, second implies there has to be a first. So the first message is the signal that binds to the receptor. Okay, so the first message is the signal that binds to the receptor. A second messenger is something that's produced or released on the inside of the cell in response to the signal binding to the receptor. So then this would be the second message on the inside of the cell. Okay, so a second messenger is going to be a signal that's produced on the inside of the produced or released on the inside of the cell in response to the first signal binding to the receptor. And just to remind you, I gave you, I think, 4 examples of second messengers. So calcium ions were one example, diacylglycerol, inositol triphosphate, and cyclic AMP was the 4th one. So those are all listed on the board and spelled out, or on the slide and spelled out for you. Does anyone remember the advantages of second messengers in signaling pathways? So there were 2 specific advantages for having a second messenger as part of your signaling pathway. Okay, so the 2 advantages, I know this was a long time ago, well, relatively long time ago. So the 2 advantages were, one was speed. So most second messengers are small molecules, which means they diffuse quickly through the cytosol or cytoplasm. So they increase the speed of the signal. And then also they amplify the signal. So speed and amplification. So what happens in the case of second messengers is let's say you have one receptor that gets activated, that doesn't lead to one second messenger. It leads to many second messengers either being released or formed. So you go from one activated receptor to many second messengers, and then each of those second messengers activates the downstream part of the pathway.
Is there always a second messenger?
No, not all. So we went through 2 examples of signaling pathways that involved second messengers, and both of the ones that we went through involved G protein coupled receptors. We did also go through an example of a signaling pathway that didn't have a second messenger, but it involved a protein kinase cascade. Okay, so another thing that we talked about were different types of signaling that can occur when you're talking about signaling in a multicellular organism. What type of signaling is it called when a cell secretes a growth factor that binds to a receptor on neighboring cells, causing them to proliferate? This is an example of what type of signaling? So you have a signal released by one cell, and that signal acts on a neighboring cell. No, not contact dependent. So in contact dependent, the signal is not released from the cell. It's bound to the surface of the cell. And so in contact dependent, you have the signal bound to one cell, the receptor bound to another cell, so the two cells have to touch each other in order for the signaling to take place. So in this type of signaling, the signaling molecule from one cell is released from that cell to the outside. It travels and binds to a receptor on a neighboring cell and then activates that receptor.
Is it deep?
Right, deep paracrine signaling. So it'd be paracrine signaling. So number 20, when a cell secretes a growth factor that binds to receptors on its own membrane, preventing it from proliferating, this is an example of, so this is talking about the cell that releases this signal, the signal that's released by the cell binds to a receptor on the same cell. So what type of signaling is that? What's that? Right, E, autocrine signaling. So autocrine signaling is when a cell releases a signal and then that signal ends up binding to a receptor on the same cell. Okay, 21, hormones are released from one cell and act on other cells and distant organs, and thus are an example of what? Right, C, endocrine signaling. So remember, endocrine signaling involves hormones, and also if you're talking about endocrine signaling in animals, it also involves the bloodstream. So the hormone gets released into the bloodstream and travels to a distant site in the body, leaves the bloodstream, and acts on its target cell. Okay. So 25 and 26 are kind of linked, and we'll go through 25 and then I'll explain how they're linked. So 25 says if epinephrine increases glycogen metabolism in muscle cells, it would have the same function in brain cells. No. Okay, so one of the things that we talked about is that different cell types can respond to the same signal in different ways. So the same signal in your body can be, different cell types within your body can respond to the same signal in different ways. And then there were three reasons that I gave you why a cell could respond in different ways, and that's what 26 is getting at. So insulin can activate lipid synthesis in adipose cells and block breakdown of glycogen in livers, so they both respond differently. And then they're asking how could one hormone have different effects on different organs, and you want to check all the ones that could apply to leading to this difference. So how about the first one, that adipose and liver cells express different insulin receptors? Yeah. So one of the things we talked about is the fact that there are many signals that have not just one receptor, but several different types of receptors in your body. And a cell's response can depend on what type of receptor it's expressing. Okay, how about the second one? Adipose and liver cells may have unique signal transduction pathways activated by insulin. Yeah. So the first thing that can be different is the receptor. The second thing that can be different is what's the signal transduction pathway that's activated on the inside. So that can also be different. I'll just go off because so far no one's gotten this one. So the next one is not true. So adipose cells do not express the insulin receptor while liver cells do. In the question, they told you that both cells respond, so they both have to have expressed a receptor. So one of them can't not have a receptor. And then the last one, how about adipose and liver cells may contain different transcription factors that regulate the expression of different genes? That's true too. So the last thing that could be different about, that could lead to two different cell types responding differently is that the effector proteins, so remember those are the proteins that carry out the response of the cell. If in different cell types you can get different effector proteins and therefore different responses in different cell types. So in this case they're saying you have in each cell type unique transcription factors that are being activated, and that's going to lead to unique genes being expressed in each case. Okay, do you guys have any other questions that you want to go through from this topic? Okay, so let's get started with photosynthesis. We're going to start photosynthesis, before we get into photosynthesis, we're going to talk about two things, but just as an overview of what we're talking about in photosynthesis is we're talking about how an organism can take light energy and use that light energy to make organic molecules that would include food molecules for that organism. Okay, so we're going to talk about how light energy is absorbed, how it's used then to make carbon-containing molecules, including food molecules for the organisms that are capable of photosynthesis. Before we get into photosynthesis, I want to talk about this important cycle. So the cycle is on the right here. This is an important cycle that exists in nature, and the existence of most organisms, depends on this cycle. So meaning that their life depends on this cycle happening. Okay, so it's a cycle that exists between photosynthesis, which we're talking about today, and then aerobic cellular respiration, which is the next topic that we're going to talk about. Okay, so we're going to talk about this half today. So in photosynthesis, you're going to take and absorb light energy. You're going to use carbon dioxide from the atmosphere and then also water from the environment. And you're going to use those three things to make organic molecules, including food molecules, and then as a byproduct you get oxygen. Then we're going to see that the food molecules and the oxygen that you make in photosynthesis are used in cellular respiration or aerobic cellular respiration to make ATP for the cell. And then as byproducts you get carbon dioxide and water, and these products of aerobic cellular respiration can feed into photosynthesis. So that's just a cycle that exists. So the products of photosynthesis feed into cellular respiration, and then some of the products of cellular respiration feed into photosynthesis. And again, the existence of most species on Earth depends on this cycle that exists between these two processes. Okay, but in terms of photosynthesis, the different types of organisms that are able to perform this include plants, algae, and then certain bacteria. If you remember back to cell structure and function, you actually looked at two what are called cyanobacteria, and those cyanobacteria are bacteria that are capable of performing photosynthesis. In this class, we concentrate on how photosynthesis takes place in plants and algae, and therefore we're going to talk about chloroplasts, and we will pretty much leave alone talking about how photosynthesis in bacteria takes place. Okay, the second thing that I want to go through before getting into photosynthesis is just to remind you of something we've already talked about, and that's the difference between autotrophs and heterotrophs, simply because the organisms that are capable of photosynthesis are a certain kind of autotroph. So remember, an autotroph is an organism that's capable of making its own food. Okay, so an autotroph is an organism capable of making its own food. And then if you get the energy to make your own food from light, then you're called a photoautotroph. So plants, algae, and cyanobacteria are specifically photoautotrophs. If you go on to take 1114, you'll learn about chemoautotrophs. And chemoautotrophs use inorganic molecules in their environment as their energy source to produce organic molecules. Okay. And then heterotrophs, in contrast to autotrophs, are not able to make their own food, and so they're going to have to acquire organic molecules from other organisms. And in terms of heterotrophs, all species of fungi and animals are heterotrophs, and then most bacteria and protists are also heterotrophs. Okay, so now that we got that out of the way, we're going to move on now to talking about photosynthesis. And the first thing we're going to talk about is the specific cell type that are the primary sites of photosynthesis in most plants. And the name of the cells they're called mesophyll cells. And you can find these cells in a leaf. And this is showing you a cross-sectional view of a leaf. And you have a top, I'll just say top and bottom layer of epidermal cells. Those are on the surface. So those would be exposed to the external environment. And then sandwiched between those two epidermal cell layers are your mesophyll cells, and the chloroplasts within those cells are the primary sites of photosynthesis in most plants. Okay, so that's just describing a little bit about where it's going to take place in a plant. Specifically within those cells, it's going to take place in chloroplasts. And when we talk about the different reactions that are a part of photosynthesis, we're going to be referring to specific membranes and specific areas of the chloroplast. So what we're going to do now is just review the structure and areas within a chloroplast. So just to remind you that a chloroplast has three membranes. So it has an outer membrane, an inner membrane, and then also a thylakoid membrane. Okay, so an outer membrane, an inner membrane, a thylakoid membrane. And then remember, the thylakoid membrane forms these disc-like structures that are called thylakoids. Okay, so the disc-like structures are called thylakoids. And then the space on the inside of a thylakoid is called the thylakoid lumen. So this is supposed to be pointing to the inside of a thylakoid. That's the thylakoid lumen. It's the space that the thylakoid membrane surrounds. The next thing is not so much important for our discussion of photosynthesis, but you can have thylakoids within a chloroplast stacked on each other. And a stack of thylakoids, if you remember, is called a granum. But we're not going to really refer to that at all during photosynthesis. But then the next thing is important is another region of the chloroplast that we talked about, which is called the stroma. And the stroma is the region of the chloroplast between the inner membrane and the thylakoid membrane. Okay, so again, the stroma is the region of the chloroplast between the inner membrane and the thylakoid membrane. Okay, so important for our discussions today is going to be the thylakoid membrane, the thylakoid lumen, and the stroma, because all those areas are going to be involved in the different reactions that we're going to talk about. Okay, so in terms of the equation, this is an equation that summarizes photosynthesis in green plants when glucose is the product. You don't have, I won't ever ask you to write it out, but you should be able to recognize it in a multiple choice question. Okay, so you should be able to recognize this equation. So this equation, again, summarizes photosynthesis. So this is a process that takes carbon dioxide along with water and light energy and synthesizes glucose, and then as byproducts you get oxygen and water. Okay, but the main goal is making this organic molecule here for the plant. That's the main goal of photosynthesis. Okay, so photosynthesis is going to include several steps. So the first step, you're going to have to capture the light energy. Then the second step is you're going to use that energy that you captured to produce two important molecules, ATP and NADPH. And the molecules that you produce in the second step are important because they're used in the third step along with CO2 to actually make the organic molecules for the cell. Okay, so you're going to use the ATP and NADPH along with CO2 that you take up from the environment to make organic molecules for the plant. Okay, but when we talk about photosynthesis, we're going to divide it into two stages. And the first stage is going to be called the light reactions. And the second stage is called the Calvin cycle. Okay, so those are going to be the two stages. And just to connect that with the steps that we just talked about, the first two steps, so capturing light energy and then using it to produce ATP and NADPH, that's both part of the light reactions. Okay, so the light reactions will include steps 1 and 2, and then step 3 is the Calvin cycle. Okay, so just to connect that with what we just talked about. Okay, so in order to accomplish these different steps, I'm going to talk about the different things that are directly responsible for them. So the first thing is you're going to have something that actually captures the light energy, and that's going to. And in plants, we have two. Plants have two photosystems. One called photosystem 1, and the other called photosystem 2. Okay, and so their job, their role in photosynthesis is to capture light energy. Okay, so these are the complexes that capture light energy. These complexes contain chlorophyll. So the chlorophyll that's present in a plant is a part of these photosystems, and they're the part of the photosystems that actually absorb the light energy. Okay, both of these photosystems are found in the thylakoid membrane. Okay, so they're both embedded in the thylakoid membrane. And then in the next step, we need to produce ATP and NADPH. ATP is going to directly be produced by an enzyme simply called ATP synthase. NADPH is going to be produced when NADP plus is reduced to NADPH, and that's going to be performed by an enzyme called NADP plus reductase. Okay, and ATP synthase is embedded in the thylakoid membrane, and NADP plus reductase is associated with the thylakoid membrane. So they're both located at the thylakoid membrane. So ATP synthase and NADP plus reductase. Okay, and then the third part is actually it's a circular pathway that's responsible for it that we call the Calvin cycle. So this is going to consist of a series of enzymes that are ordered in a circular pathway, and this cycle takes place, all these reactions take place in the stroma. Okay, so on the next slide, at the bottom of the slide, I'm asking, do the two stages happen in the same place? So if you use what I just told you now, you should know that they do not happen in the same place. So the light reactions happen at the thylakoid membrane. So I'm just going to write TM next to light reactions. And that's because all the components that are involved in light reactions are either embedded in that membrane or associated with that membrane. Okay, so the light reactions happen at the thylakoid membrane. And the Calvin cycle occurs in the stroma again. Okay. So when we split it up into these two stages, you can think of the light reactions as the photo part of photosynthesis and the Calvin cycle as the synthesis part. So this just gives you a large overview of what's going to happen. So we're going to see in the light reactions, that's when you capture light energy, use it to make ATP and NADPH, and you also, as a byproduct, produce oxygen. So oxygen is a waste product of photosynthesis, but it's important for us, obviously, but it's a waste product of photosynthesis. And then ATP and NADPH are really important because the Calvin cycle depends on them. So the Calvin cycle will not take place without the light reactions because it needs ATP and NADPH. So then the Calvin cycle is going to use those two things produced by the light reactions along with carbon dioxide to make a 3-carbon sugar called glyceraldehyde 3-phosphate. And this is the first important product of the first important organic molecule that's produced by the Calvin cycle, and then the plant can use that glyceraldehyde 3-phosphate, which again is a 3-carbon sugar, to make other types of organic molecules. So that's kind of like the building block for other types of organic molecules. But the glyceraldehyde 3-phosphate's the important product of the Calvin cycle. That's what you're actually trying, that's the goal of that cycle, is to produce that molecule, and then the plant can use that molecule to make other organic molecules, including glucose, cellulose, starch, etc. Okay, so those are an overview of the two things that, the two stages. So the first thing we're going to talk about is what form of light energy is important for photosynthesis. So what is being shown on the slide here is the full electromagnetic spectrum. So you have gamma rays, X-rays, UV light, visible light, infrared light, microwaves, and radio waves. So that's every possible wavelength of electromagnetic radiation. The portion of the electromagnetic spectrum that's important for photosynthesis is visible light. Okay, so that's the light energy that's absorbed during photosynthesis. And this range is simply called visible light because that's what we can detect with our eye. Okay, so visible light is the portion of that spectrum we can detect with our eye, so it's called visible light. And that's the light that's going to be driving photosynthesis. Okay, you also then are going to have something that captures this visible light, and we already said that the role for capturing that visible light is performed by photosystems, and plants have two of them, photosystem 1 and photosystem 2. And these are complexes of both pigments, and chlorophyll is the main pigment in plants, and proteins. So photosystems are complexes of proteins and pigments. And we're going to talk about pigments first because that's the part of the photosystem that actually absorbs the light energy. Okay, so a pigment you can define as a molecule that can absorb light energy. And then, as I already told you, the main pigment in plants is chlorophyll. There's two versions of chlorophyll. There's A and B. Both versions of chlorophyll are going to absorb red and blue wavelengths of light. So in terms of what range of visible light they absorb red and blue, but they reflect green. And that's why plants appear green, because what you see is the color that's reflected by the pigment. Okay, so, and there's other pigments that plants have that absorb other wavelengths of light while reflecting others, but because chlorophyll is so abundant, it masks the presence of those other pigments that may be present in the plant. Okay, so the chlorophyll molecules that are part of these photosystems are important for capturing the light energy. And what we're going to talk about now is what happens to a pigment molecule or a chlorophyll molecule when it absorbs light energy. And what happens is an electron is going to be boosted to a higher energy level. Okay, so an electron within the molecule is going to gain energy and be boosted to a higher energy level. And they're calling this the excited chlorophyll molecule. So that's the chlorophyll molecule with the higher energy electron. And then what we're going to talk about now is two things that happen in photosystems in terms of what happens after you've excited that chlorophyll molecule. Okay, and what we're going to talk about in a second is that photosystems consist of two functional parts. So photosystems consist of two functional parts. They consist of an antenna complex and a reaction center. And we'll talk about what both of these do with respect to the photosystem. Okay, but those are two functional components of a photosystem, the antenna complex and the reaction center. And in terms of what's down here, okay, you have three options, 1, 2, and 3. 2 is what happens in the antenna complex. Okay, so 2 is what happens in the antenna complex, and 3 is what's going to happen in the reaction center. Okay, so in the antenna complex, what's going to happen is you have a bunch of chlorophyll molecules, one chlorophyll molecule is going to absorb the light energy, become excited, and then what it's going to do is pass the energy to a neighboring chlorophyll molecule. When it passes the energy, its electron goes back down to its unexcited state, and it boosts the energy of the electron in the other chlorophyll molecule. So in the antenna complex, what's going to happen is the pigment molecules are going to be passing the energy that they absorb from pigment molecule to pigment molecule. But you're only passing the energy, you're not passing anything else. Okay? Then what's going to happen is the energy is going to reach the reaction center, and you want to pay attention to the green molecule here. What happens is when the energy excites this chlorophyll molecule, this chlorophyll molecule actually gives up that electron. Okay, so rather than just passing the energy, it gives up the whole electron to another molecule, and then it has to be replaced by another electron, and then it has a lower energy electron that replaces the high energy electron that it just released. Okay, so again, in the antenna complex, you're just going to be passing energy between molecules, and then in the reaction center, you actually get the release of a high energy electron, and then we'll talk about what happens with respect to that electron in the light reactions. Okay, so previously mentioned that photosystems can be broken down into two functional components. So that's the antenna complex and the reaction center. The role of the antenna complex is twofold. One, it's the part that actually captures the light energy. Okay, so the antenna complex captures the light energy, and then it funnels it to the reaction center. Okay, so again, the antenna complex captures the light energy and funnels it or channels it to the reaction center. And then what happens once the energy reaches the reaction center produces, and by produce I mean releases a high energy electron. And then we're going to see where does that electron go once it's released from the photosystem. Okay. So again, antenna complex absorbing light energy, important for absorbing the light energy and funneling it to the reaction center. The reaction center responds by producing a high energy electron. And so the electrons that we're going to be talking about are going to be being released from the reaction center. Okay. So now we're kind of ready to talk about the light reactions. And the light reactions are going to involve the following components. What we're going to do is kind of put them in order and talk about how they interact. So it's going to involve ATP synthase and NADP plus reductase, which we already talked about. And then it's going to involve two photosystems, 1 and 2, and then 2 electron transport chains. Okay, so what I'm going to do is first draw on the board what happens in terms of the light reactions, and then I'll go back to using the slides to re-describe what happens during the light reactions. Okay, so we're going to start, again, we're talking about the light reactions. And the first type of electron flow that we're going to talk about is non-cyclic electron flow. And that's obviously going to be compared to cyclic electron flow. Okay, so we're talking about the light reactions, and the first pathway of electrons that we're going to talk about are going to be non-cyclic electron flow. Okay, so this pathway is going to start actually with photosystem 2. Okay, the reason that they're named 1 and 2 is they were named in the order that they were discovered. So photosystem 2 was just discovered second, so they named it 2, but 2 actually acts first in this pathway. Okay, so what happens is photosystem 2 absorbs light energy, again that happens by its antenna complex. The antenna complex funnels it to the reaction center, and then photosystem 2 spits out a high energy electron. Okay, so absorbs light energy, releases a high energy electron. Now photosystem 2 can't just keep on releasing high energy electrons, it needs to replace them, so it needs something to give it electrons. And photosystem 2 gets electrons by splitting water. So it takes water, splits it in half, and takes electrons from it, and those electrons replace the electrons that are leaving photosystem 2. And when you split water, you end up producing oxygen and this is for 2 water molecules, and some hydrogen ions. Okay, so we actually produce oxygen, which is a waste product again of photosynthesis in this first step of the light reactions, and it's produced by photosystem 2. Okay, and then what I'm going to do here is put in here, this is going to be the thylakoid membrane. Okay, and the thylakoid membrane separates the stroma from the thylakoid lumen. And so in my drawing, the stroma is going to be up here, and the thylakoid lumen is going to be down here. Okay, these electrons that are released by photosystem 2 are going to be passed to the first electron transport chain. And just to quickly describe what an electron transport chain is, so an electron transport chain is going to consist of a series of components, and the different components just pass electrons in a specific order. So component 1 passes the electrons to component 2, 2 passes to 3, and then however many components there are, that just keeps on going. Okay? So in an electron transport chain, you're just going to be passing electrons from component to component. So the electrons get passed to this first electron transport chain, which consists of several components, but I'm just drawing it as one, just to make it easier. Yes?
So does the antenna, that's what it's called, right?
Antenna complex?
Yeah.
Yeah.
Does that go through the outer membrane? Like how's it getting light energy if it's inside of the
Well, the light energy can pass through the membranes.
Oh.
So, yeah. So the light energy is able to pass through the membranes. Yeah. Okay, so you do need to know one component of electron transport chain number 1, and that component is going to be cytochrome B6 F complex. So it's got a rather complicated name, but it's called the cytochrome B6 F complex. So again, this complex is just one component of this first electron transport chain. Okay, I can put this down. Okay. Oh, I didn't know it was touchscreen. Okay. Okay, so the reason that you have to know this part, this component, is because as electrons pass through this complex, it pumps hydrogen ions into the thylakoid lumen. Okay, so this complex pumps hydrogen ions into the thylakoid lumen. Okay, and this is going to help create a hydrogen ion electrochemical gradient that's going to be important later on. Okay, so I'll say that again, but this is going to help to create a hydrogen ion electrochemical gradient that's going to be important later, actually at the very end when we're done with the pathway. Okay, so as electrons pass through this first electron transport chain, cytochrome B6F complex pumps hydrogen ions into the thylakoid lumen, and then the electrons are going to leave this electron transport chain number 1, and now they're going to be passed to photosystem 1. Okay. And then what happens is photosystem 1 also absorbs light energy. So what happens is the electrons that are arriving at photosystem 1 are going to gain more energy as photosystem 1 absorbs light energy. Okay? So again, the electrons are going to gain more energy, and then that means photosystem 1 releases a high energy electron. And then those electrons are passed to a second electron transport chain. So this will just be ETC number 2. You don't need to know any specific components of this electron transport chain. And then at the very end of this electron transport chain, the electrons are passed to the enzyme NADP plus reductase. And what NADP plus reductase does is it takes the electrons and it's going to use the electrons to reduce NADP plus to NADPH. Okay. So ultimately, water in this process is the electron donor. So that's water's role. Remember, it was part of one of the reactants in the equation that summarizes photosynthesis. So water is important because it's the electron donor. And the electrons that come from water in this pathway ultimately end up in a molecule of NADPH. Okay, so they're going to flow from water and then ultimately end up in an ADPH.
Does that last hydrogen electron from electron chain 2 go to the NAD plus reductase or no? Does it just only go through the process of NAD plus?
No, well, NADP plus reductase catalyzes this reaction.
So the electron's still used?
Yes, yeah.
So do the I guess this is kind of technical, but do the electrons need to be more powerful, or does it just help speed up the reaction?
Well, so what happens is, in terms of this first electron transport chain, the electrons are going, as they flow from 2 to 1, they lose energy, but some of that energy is used to do this. And then the high energy electrons that are added in NADPH are going to be used for reduction in the Calvin cycle. So their NADPH takes the electrons, carries them to the Calvin cycle where you use them to reduce an organic molecule. Okay, so so far we've produced 2 of the products that we said in the light reactions. We've produced oxygen, which we said was a waste product, and then we've produced NADPH, which is an important product for the Calvin cycle, and then the other product that we mentioned was ATP. And this we said was going to be formed by, or produced by ATP synthase, which is also in the thylakoid membrane. And ATP synthase is going to catalyze this reaction. It's going to catalyze the synthesis of ATP. So it's going to take ADP and an inorganic phosphate and attach it to ADP to make ATP. And if you remember back to energy enzymes and metabolism, we said that that reaction is endergonic, which means that it requires energy. So this enzyme requires an energy source. Okay? In order for it to catalyze that reaction, it's going to require an energy source. Okay? And the energy source is the hydrogen ion electrochemical gradient. Okay? So a hydrogen ion electrochemical gradient is the energy source. And there's several things contributing to this hydrogen ion electrochemical gradient. You're producing hydrogen ions here in the thylakoid lumen. You're pumping hydrogen ions into the thylakoid lumen here, and then you're going to be using up hydrogen ions in the stroma, and so all of that together effectively concentrates hydrogen ions in here in the lumen relative to the stroma. So if hydrogen ions are allowed to flow down the electrochemical gradient, they're going to flow from the lumen into the stroma. And what ATP synthase does is it provides a pathway for the hydrogen ions to move through it. And it's the flow of hydrogen ions through this synthase. So hydrogen ions are going to flow through this synthase from the lumen into the stroma. And that's what powers this synthase. So similar to the fact that you can use water flowing down a waterfall to produce electricity, you're using hydrogen ions flowing down their electrochemical gradient through the synthase, and that cranks the synthase and allows it to produce ATP. So the energy source, again, is this hydrogen ion electrochemical gradient, and that chemical gradient is created during this electron flow that we just talked about. that's a good point. So keep this separate. This is never going to receive electrons. ATP synthase has nothing to do with the electrons. It's not a part of the electron flow. The connection between the electron flow and ATP synthase is the hydrogen ion electrochemical gradient, which is created during electron flow and then used by ATP synthase. Okay, so we're going to do the same thing, and I draw it out slightly differently, but I'll explain how it's different on this slide. But basically, we're taking all these components, which you've already seen, and going to put them in the correct order. So in this drawing, this double blue line is the thylakoid membrane. So the double blue line is the thylakoid membrane, and again, that separates the stroma at the top from the thylakoid lumen. And remember, our pathway is going to start with photosystem 2, which is going to absorb light energy. In response to that light energy, it releases a high energy electron. And then remember, it replaces that electron with electrons that it gets from splitting water. And when it does that, it produces oxygen, hydrogen ions, and then it also steals the electrons that it needs to replace the ones that are leaving. Then we said that these electrons being released by photosystem 2 are going to be passed to the first electron transport chain. That is not going to be drawn on this slide. So what is drawn on the slide is the CBFC is cytochrome B6F complex, which was the one component of the first electron transport chain that I wanted you to remember. So the reason I have it drawn in this picture is because as electrons pass through this complex, it pumps hydrogen ions into the thylakoid lumen. The electrons leaving the first electron transport chain are then going to be passed to photosystem 1, which absorbs light energy and then releases a high energy electron. And then the electrons leaving photosystem 1 are passed to a second electron transport chain, which is not drawn on the slide. And then we said at the very end of that electron transport chain, the electrons are passed to NADP plus reductase, which uses them to reduce NADP plus to NADPH. Okay, so far we've produced, again, oxygen and NADPH, and we've also created a hydrogen ion electrochemical gradient. And then we said that the hydrogen ion electrochemical gradient is going to be the power source for ATP synthase to power the production of ATP. And specifically, it's the hydrogen ions flowing through the synthase that is powering the synthase. Okay. Do you guys have any questions about, so this again is the light, this is all of that just alone was the light reactions. And specifically that was non-cyclic electron flow. No. No. So you missed it. I'm only going to go till 8. So I'm almost done. Because my other classes only got this far. So.
You said the hydrogen ion electron gradient is you only use for the ATP, but it could use it to power the ATP.
Yeah. Mm-hmm. Okay, so this is just a picture taken from your book, and you can use it if you find it useful, or just ignore it if you find it confusing. It's just the light reactions that we just went through, and it's got the different components like photosystem 2, 1, the cytochrome B6F complex, NAD plus P plus reductase, and ATP synthase, all kind of embedded in the thylakoid membrane. If you find it useful, use it. If you don't, just disregard it. So what we just again described is non-cyclic electron flow, and non-cyclic electron flow produces oxygen, NADPH, and ATP. Okay, so non-cyclic electron flow produces 3 things: oxygen, NADPH, and ATP. And we said that 2 of those things are important for the Calvin cycle: the ATP and the NADPH. Okay, so the ATP and NADPH that are produced are necessary for the Calvin cycle. Okay, yeah.
Quick question. So how come, like, photosystem 1, is there a reason why it's, like, in the bilayer? I mean, I see, like, photosystem 2 releases, it has, like,
Photosystem.
H2O and then oxygen and hydrogen. Does photosystem 1 not give anything off? Like, is there a reason why
They're both embedded in the membrane, but only photosystem 2 splits water. Photosystem 1 can't do that.
Okay. So it's just there just to collect light then?
Yes.
Okay.
Yeah.
That's it. All right.
Okay. So again, the ATP and NADPH that you produce are used for the Calvin cycle. What we're going to learn is that the Calvin cycle actually uses more ATP than it does NADPH. Okay, so it uses more ATP than it does NADPH. So you could have a situation where you're making not enough ATP and too much NADPH. Okay? And some plants have a pathway for producing only ATP without producing more NADPH. And that pathway is called cyclic electron flow. Okay? So we're going to talk about cyclic electron flow, and cyclic electron flow has one product, ATP. Okay? So cyclic electron flow does not produce oxygen and does not produce NADPH. It only produces ATP. So this would be a pathway that the plant could use to produce ATP without having to produce more NADPH. Okay, and there's two components of this flow that you're going to have to remember. That's going to be photosystem 1 and then the cytochrome B6F complex, and then you just have to know the path. Okay, you don't have to know the other components that are drawn on the slide there. So this path is going to start at photosystem 1. Okay, so cyclic electron flow starts at photosystem 1 rather than photosystem 2. So photosystem 1 absorbs light energy, releases a high energy electron. This electron that leaves photosystem 1 gets passed eventually to the cytochrome B6F complex, which pumps hydrogen ions, creating that hydrogen electrochemical gradient that's powering ATP synthase. And then after the electrons pass through the cytochrome B6F complex, they ultimately get passed back to photosystem 1. So it's called cyclic because they end up where they began. They start at photosystem 1 and they end at photosystem 1, and during that cycle they are going to pass through the cytochrome B6F complex. So cyclic electron flow only involves photosystem 1. It does not involve photosystem 2. And in this pathway, the electrons are never passed to NADPH. They're just cycling around in that circular pathway. I don't know. I don't know the answer to that. I don't know if it's known. Okay, but then when you compare the two, cyclic to non-cyclic, it's important that you know the difference in the products. Okay, so non-cyclic, which is on the board, produces ATP, NADPH, and oxygen, whereas cyclic only produces ATP. Okay, so that's where we're going to stop for today, and where we're going to pick up next week is with talking about the second stage, which is the Calvin cycle. And in this stage, we're going to see how the ATP and NADPH that we just produced are used to make 3-carbon sugar. Yep.
So what's the purpose of even having the first and the second electron, like the photosystem 1 and the electron transfer thing? Couldn't it just go to that?
So can you say again?
Sorry. Why does it, if it's just the PS1, if that only collects light, why does it need to be light energy?
Well, I mean, that probably is not what you're looking for, but it connects electron transport chain number 1 to electron transport chain number 2. So it's part of the transfer of electrons. So without it, these electrons wouldn't be able to go from here to there. And why it evolved that way, I don't know. So why you need all these different components, it's just how it works.
So you're saying it just couldn't start in photosystem 1?
What?
It couldn't start in photosystem 1. It has to start in photosystem 1.
No, cyclic electron flow can start with photosystem 1, but non-cyclic always starts with photosystem 2. Okay, I'll send you guys the announcements tomorrow morning during my office hour.